Good morning, everybody. Everyone being Christina, just you and me. Ah, it's like one-on-one tutoring. Yeah. I don't know what my cats are doing. I was just about to show you them. Hopefully a few more people will show up, but if not, we'll crack on anyway. So, let's get everything shared up. Okay, there we go. Ah, it's Tanya as well. There's now plurality. There she is.
Good morning.
I wonder if I've scared everybody off from the last exam. All right, so
Dr. Crook, do you know what was like the class average to see how bad I was?
You're at or near the top for that exam. It was, I think it caught people by surprise. I'm not really sure why. So I tried kind of stressing how important it was to kind of get the details and the concepts at the same time, and that the fact that there'd be a lot of stuff in there because we did cover a lot of different stuff. And it was kind of weird because in both sections of this course that I'm teaching, the Monday, Wednesday, Friday, and this one, people are doing really well in the first two exams. Not everybody, but a lot of people are doing really well, surprisingly so, actually, given my experience from teaching this other semesters, even online. And the grades tanked for the third exam. Really, 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 really badly. Like people went from 100% down to 30%, which to me just doesn't make any sense. Because if you're able to get up around 90 to 100%, then even if the exam's tough, then you should still be getting, 75 to 85%. And people, it really tanked. So I don't know really what happened there. It's a bit of a mystery, to be honest. So that's why I don't think an average would be particularly informative for you, but you did well. And again, I mean, you did. Sorry, dog's coming downstairs. The exams are hard, but the course as a whole is designed so that as long as you're putting the effort in all the different ways, the homeworks, the lab, and the exams as well, you can still get a really good grade because the homeworks and the lab is deliberately designed to pull your grade up so that exams are not the only way in which you're being assessed. Right. So if you do have a less than stellar exam result on one of your exams, it's not going to crucify you for the course. I mean, that's very, very deliberate, very intentional. And so, you know, people can be getting like bumping around on DC average in the exams, and they can still potentially get a C in the course if they're doing all the other stuff that they need to do. So.
I don't know. I think well, I went over the answers and I think what I got a lot wrong were the partial points, like the click all that apply, because I didn't put one, but I'm always kind of afraid because if I click more than what it is, I'm gonna get deducted. But if I didn't, I'm gonna get deducted. So it was a lot of like detail, get it like straight detail. And I think that's what I messed up because I have like a lot of partial points.
Right. Well, and in some ways, that those multiple answer questions are two-edged sword, right? So when they were like on a Scantron in an in-person class, you know, those questions would be designed so it's either right or wrong. And so you either get 5 points or you get no points. Whereas the partial credit at least gives you some points towards that. But the flip side is that you have to really know your stuff to get all of them, right? Because again, there's that risk of penalty, and the penalty isn't huge, right? It's not designed to be punitive. It's designed to stop people just ticking all the boxes on every single multiple answer question, right? Because I mean, that's an easy strategy if there's no consequences to doing that. So yeah, the penalties usually altogether, all the not correct choices are usually a little bit less or a little bit more than one of the correct answers. So, you know, it's still a gamble, but you're not.
That misses my insecurity because I took a long time. I was like, oh, is that right? Am I missing something? And it
Right.
ended up I was missing, but it was just like insecurity.
Yeah,
Those
and
are bad questions.
I'm sorry. I mean, it's not designed to do that or to elicit that response. Um, but as I say to any students, no matter how good or bad, insecurity comes from not knowing the material as well as you think you do. That's always the issue. And it's I so many times have students say, oh, I thought it was this one, but then I looked at that one and I thought that one looked kind of right, and I kind of chose the right one, and then I crossed it out and I went back and chose the wrong one, right? And some of the questions do have answers that kind of look right superficially, right? So if you just took a quick glance at them and didn't really think about it, it'd be like, oh, that could be right, but they're not. And so that isn't, they're not there to trick you, right? They're not kind of like gotcha sort of answers. What I'm doing quite deliberately, because I'm very deliberate about all of this, is I'm seeing how well you really know the material. Because if you kind of know it and you have the general overview, but you're short on some of the details, then some of those answers can start looking kind of about right, but they're not. So really what I'm doing is testing how really truly do you understand that material? Because if you really truly do, then it's quite obvious there's going to be something in one of those answers, even if it looks about right, that when you read it through again, you're like, no, that doesn't fit. I know that's wrong because this, and this is right, but that is definitely not right. So this whole answer is wrong. Right. So, and that's,
Right. And
yeah.
you were correct. I was regretting because I was like Dr. Crick mentioned a lot, like study this. This is gonna be like it's hard, study the material. And I was like, oh, okay, I kind of know it. So and I surely was not when I took the exam. I was like, I needed more studying.
Yeah, and this section is going to be pretty much the same, right? Because I mean, look at this, like on the screen in front of you. This is a fairly straightforward example of transcript of gene regulation in prokaryotes. Sounds like my cat's throwing up somewhere, or one of them anyway, right? And you just look at that, you're like, holy crap, that's complicated, right? And this is the simple one, you know. So, and we have a bunch of time to go through this. Oh, cramps, where's the? Here we are. Right, so we're going to be doing this today and Thursday, right, for this one PowerPoint. And, you know, we're going to go through it thoroughly, but there's a lot of stuff here. And it can get, it gets pretty tricky because you've got to tie your head into different knots to understand what's going on. This one's relatively straightforward. This is the lac operon, basically. We're going to be studying two different systems in prokaryotes. One's going to be the, where is it? Not that one. Here we go. The lac operon, right? And so, you know, all the times I said that prokaryotes do their thing in a funky way, but they are pretty cool about how they control their gene expression. These are really good examples of that, right? They're actually fairly complex, right? So this is the lac operon. And where's the trip operon? This is the trip operon, which has an extra funky thing going on, which is called termination, right? So it's not a trivial topic to study, right? And only gets more complicated when we talk about eukaryotes. Not necessarily because of what they do is more complicated, but there's more ways in which you can do it, right? But also, like the reason for spending what's going to be, what, 2 whole weeks on this, which is really the bulk of what's left of the course, is that this is how organisms are the way they are, both in an evolutionary sense, right? How they came to be what they are, and also how they deal with their environment, either internally, like us, we're multicellular organisms, where we have an awful lot of homeostatic processes like temperature, blood glucose, blood osmolarity, all that kind of stuff, right? Which we do without even thinking about it, thankfully, because it'd be really complicated otherwise. But also in terms of our external environment as well, right? So we're really, actually, if you think about it, I'm sitting underneath a fan, I'm fairly comfortable temperature-wise. We're very good at modifying our environment to suit us. It's kind of like external homeostasis in a sense, right? The whole joke about how no one lived in Florida until air conditioning was invented because it was such a miserable place to be. It's not wrong, right? There's some truth to that. Same for Texas, I imagine, to some extent too. But an awful lot of other organisms don't, right? So how do they cope with their environment when their environment changes? And for example, the two examples we're going to look at in bacteria are all about nutrition, right? How do bacteria can essentially take advantage of different nutrients, or how do they control, what they're doing in response to nutrient levels, both internal and external, and how do they avoid doing that when they don't need to, right? And so you can think about these examples all the way through, and really both of those perspectives are relevant, like the evolutionary perspective and the everyday, day-to-day perspective, right? And even in multicellular organisms, you have something in between, which is the control of development. Right? That's one of the courses that I teach once every blue moon when I'm not teaching genetics, right? And so how do we get from a single cell to being a large, complex, multicellular organism with many specialized tissues? When every single one of those cells, every cell in your body, bar your gametes and your red blood cells, have exactly the same DNA in them. So why are your neurons up to, 3 feet long and your adipocytes, which store fat, kind of round and blobby, right? How are they such different things when they have the same instruction set, right? And so it really comes down to gene expression and control of that, right? So that's why this is something that I really enjoy teaching, because it really does start getting at biology, right? It's not just, oh, DNA replication, we've got to do that because cells divide kind of stuff. I mean, that's important, but really how all of that is controlled, how everything is controlled, really comes down to, in some part, gene sequences, but in a large part, gene regulation. And that's why it's such a huge and complex subject, because the larger, more complex the organism, then the more numerous and more complex the mechanisms to control gene expression must be. We have the same number of genes as a soil nematode that has 989 cells exactly. Someone's counted them, right? How come we are so much more complex biologically than another organism that has the same number of genes as we do, about 20,000, give or take. You know, it's partly what those genes are, right? And their difference in gene sequence, right, in terms of protein coding. But a huge, huge part of that is how do we control the expression of those genes in different contexts? And that's really what this is all about. Pardon me. So before we get on to this kind of stuff, what I want to do is put this up. Let's get rid of that. Tuck that behind there. So where are we at? We're talking about the control of gene expression. Basically, we start with DNA. That's our info, right? That's the thing that stores the information to make whatever it is that's going to be doing the stuff. And typically, but not always, let's see, oh, you can see that down there. The thing that does stuff is protein. Not always. Could also be what we call non-coding RNAs. So those, we'll get to those once we get into some of the funkier stuff later on. But we've already come across a bunch of those already. You know, we've got ribosomal RNA, we've got transfer RNA. Those are all non-coding RNAs that do things, right? But by and large, what we're talking about when we talk about control of gene expression is protein. Now, there's another little caveat here. This is functional protein. Now, you might think, well, okay, what's the difference between protein and functional protein? And actually, there's a pretty huge difference between protein, and for many proteins at least, protein that's produced by the ribosome and the protein that actually has a biological function. And we'll get into why in a little bit. So you look at this and you're probably thinking, okay, so this is pretty simple, right? We've got DNA and we've got mRNA and we've got protein, right? That's the sequence hypothesis that we learned about last section. Before we get into all of this, let's go into what's involved here, right? So this is obviously a big one. So this is all about the control of transcription, right? So you have to control access to the DNA, and then you have to start the process of transcription, right? So there's a huge amount of stuff you can do to fine tune how much mRNA is produced. And typically when people think about control of gene expression, this is the one that they think about. Well, they would if my green pen worked. No. So I didn't cover that up properly. Dang it, I'm gonna have to use blue now. Right, that's the one people typically think about when we talk about control of gene expression. It's control of transcription. They're gonna make sense too, because that's the very beginning of the process, by and large. But we also have control of RNA processing. And that's eukaryotes. And so there are funky things that eukaryotes can do with splicing in particular that will affect what mRNA you get. And then we've got a whole other bunch of stuff going on. So how long this mRNA hangs around for is going to control how much protein you get out of it at the end. So if this mRNA is very stable, you're going to be producing protein, from it for a long time to come. If you want to shut off this whole process, one way of doing so is to make sure that that mRNA does not hang around for very long. So not only do we have to turn on gene expression, but we also have to turn it off and modulate it too. So mRNA stability is really important in that process, right? And that 3' UTR and poly A tail that we spent a while talking about in the last section are really huge in that process, particularly the 3' UTR. Right, that's one of the main regions which is used to control how long an mRNA hangs around for. We've also got control of translation. Right, and both prokaryotes and eukaryotes have funky ways of doing so. In prokaryotes, it's really a lot of it's down to secondary structures in RNA, which we probably aren't going to get to today. We'll get to that on Thursday. In eukaryotes, it's quite, we learned how translation was initiated, right, by, promoted, let's say, by the 5' cap binding proteins and the poly A binding proteins. Well, if you control how that mRNA moves around, you can actually stop that poly A binding protein from interacting with the ribosome. So you're able to then control translation. What else is going on? Oh, okay. So really what we have there, and sorry if you've already drawn this out, this is kind of one of the perks of being the guy with the whiteboard. Is we have our intermediate step, right? So mRNA obviously gets translated into protein, but that's not the final end product. Sometimes it is. Kind of depends on context. All of this is actually context dependent, and we'll get into that context in a little bit. So we have protein modifications. Oh, getting into the shadow of the couch cushion there. Boofy couch cushions. And those include things like cleavage. And this is going to be awful to try and write out small. Phosphorylation. Let me type that up into the chat box because I am well aware of the shortcomings in my handwriting, particularly when I'm writing on a small whiteboard on my couch. So there are many, many, many proteins which require those modifications, particularly in eukaryotes, not solely, but especially in eukaryotes, before they become functional. And then the final one is one which isn't always super obvious, but protein degradation. Oh, actually, I should put location there, because where a protein is really important as well in terms of whether it's functional or not. So proteins, actually, even once they are functional, can be targeted for degradation. So a classic example would be a receptor, like a neurotransmitter receptor in your neuron. Once it has received a signal, like you have a pulse of signal that crosses the synapse, then that receptor typically is taken in by the receiving cell and destroyed and replaced with a fresh one. Usually they're one-shot deals. They're not able to continually sense and release the signal. Right. So, and essentially all of this, and this is like a huge kind of complex mess of information, is how we go from this to that. Right. And really, when you think about control of gene expression, how much of this you get out, this functional protein, is the sum of all of these things. And you can have processes acting together, right? So if you need a lot of protein very quickly, you're going to be boosting the control of transcription. You're going to be making your mRNA very stable. You're going to be boosting the initiation of translation and so on, right? But they can also act against each other too, right? So you might be increasing the rate of transcription, but you can counter that by increasing the rate of mRNA degradation, right? If you want to really fine tune the level of functional protein being produced, right? So really, how much of this you get out from any given gene is the sum of all of these things. And which of these matter the most, or which even contribute, depends very much on what gene you have, what gene you're expressing, and all of the context of that, right? Is it a protein that's going to be exported into the extracellular space? Is it a protein that's going to be held inactive somewhere until a signal arrives to turn it on? You know, is it something you need a lot of very quickly, or is it something you need a low level of over a long period of time, right? So it's very hard to be specific about any of these things. All I can do is really teach you the mechanisms, right? The various dials which can be turned and switches that can be flicked, so that when you come across a particular gene, you can figure out the context based on all of those principles. So I could give you examples, but for any example that I give you, all of that relative stuff is only going to apply for that one gene. And it's probably going to apply only for that one gene in that one cell under that one condition, right? That's what makes this such a complex topic to study. And I very much do my best to try and kind of dilute it down to the central principles without overwhelming you, but still giving you all the tools that you need to understand, you know, that bigger picture stuff, which isn't trivial. Okay, any questions about any of that so far? Right, because that really gets at all of what we're going to be talking about. And really, if you think about it, there's that, I mean, I've got these questions kind of listed in the PowerPoint here, and they're all very relevant, right? And particularly this one, right? If we're a multicellular organism, so obviously yeast, protists, and bacteria don't apply, but they have their own challenges too. But where is this gene going to be expressed? Clearly, in neurons, you're going to produce neurotransmitters, and those neurotransmitters are going to be specific for that neuron. So you might have a dopaminergic neuron, right, which produces dopamine. So you're only going to be producing dopamine conversion enzymes in those neurons. You're not going to be producing them in like tyraminergic neurons, for example, which use tyramine instead. And you're certainly not going to be producing dopamine in your pancreas, right? Because it has absolutely no purpose being produced there. There's no benefit. It's only cost, right? Similarly, in your pancreas and your beta cells in your pancreas, you're going to be producing insulin, right? That's one of their main products. But you're not going to be producing insulin in your muscle cells, right? Or your liver, because those are the targets of insulin production, right? So what genes are going to be turned on in which cells is a very large part of why those cells are different from each other. It's called like a cell-specific program, essentially. One whole series of genes is going to be on in one cell, and another series of genes is going to be on in a different cell. And those gene products essentially make those cells what they are. And then all of these, right? So how is the transcript going to be controlled? Or how is it the control of transcript production controlled? Its processing, its translation. Where will that transcript be, right? I mean, that's this question here really gets at least in eukaryotes, is that protein a cytosolic protein? Is it a cell membrane or plasma membrane protein? Or is it a protein that's going to be exported? And so you have to control where those proteins are produced, because that then controls where those proteins are going to end up. Right, so for example, insulin, the protein, and it is a protein, it's a fairly funky one, but the insulin protein is produced inside the rough endoplasmic reticulum, because it'll end up outside of the cell at some point, so that it can flow around the bloodstream and tell your muscle and liver cells to take up glucose. And then there's all these modifications and where that protein, and it says gene product here fairly coyly, because really it could also be a non-coding RNA as well. But generally speaking, we're talking about proteins, generally speaking, not always. But how's it going to be modified and where is it going to be localized, right? And that isn't a one-shot deal either. There are an awful lot of transcription factors, right? Proteins which control the transcription of genes, which are localized in the cytoplasm when they're not needed. One of the genes that I work on is. And so,
Dr. Kirk, for any response, any response, we have to go to gene expression and translation, transcription, gene expression to have a like a response in our body?
At some point, yes, but not necessarily directly. So, for example, you know, I just had my breakfast, which is why I'm getting a little bit, you know, sleepy, and I haven't had my coffee yet. But my blood sugar level is probably currently peaking around now, a little bit, you know, because I eat a healthy breakfast. But nonetheless, my blood sugar level is going up. But it takes hours to produce insulin. So the insulin that my pancreas is now busy releasing, because my blood sugar level is kind of getting above the comfort zone. That insulin was produced probably while I was sleeping last night. So when insulin levels drop, you know, the pancreas does, or the beta cells specifically, do need to produce more of it. But the insulin that's getting released is not getting made as it's being released. It's stored in little vesicles, basically little droplets, so that when the signal comes of a elevated blood sugar level, all those droplets are basically kind of their contents to push down to the bloodstream, which then prompts my muscle and liver to take up that glucose, and then the blood glucose level drops back down a little bit. And so that's the homeostasis part. So at the root of it, yes, everything that we do at some point comes back to control of gene expression, but not necessarily directly. So for example, you know, I can wave my arms around, I could go for a bike ride or a run or any of those things, right? And I'll be using proteins in my muscles, those actomyosin filaments, which I made, you know, potentially years ago, right? When I was growing, many years ago, actually. But if I exercise vigorously enough, I'll damage some of those, right? It's just part of exercise, essentially, you know, kind of you damage the things that you're using. And so in those cells which suffer some of that exercise-induced damage, that will turn on gene expression of the things that will fix that, either making more actomyosin filaments or making more red blood cells or whatever it might be. Do you see what I mean? So from day to day, you know, if I'm just like lying on the couch, not really doing very much, there's not a whole lot. Well, I mean, there is and there isn't, you know, but anytime you do anything or your environment changes or your diet changes or something changes in your body, there has to be some means of responding to that. And so, yeah, it's kind of, because that's getting into a lot of essentially anatomy and physiology, right? But underneath all of that are the mechanisms that will control the expression of the genes that are required for that response, in response to that particular change in conditions. You know, so things which are using stuff continually, like for example, your gut, particularly your small intestine, is continuously, or actually, you know, your stomach too, continually rejuvenating itself. Right, it's a constant process of, it's actually like a little conveyor belt. Super cool, actually, if you read into it. Your intestine consists of a series of, actually, let me draw this. It's way more fun. I'm going to take a picture of this. I just recently read an article on how the specification of particular cells is controlled, which I thought was super, super neat. And you don't need to worry about this for, you know, kind of studying and stuff. This is just a neat example to use. I typically often use different examples for different classes. And so these are things which are just for your kind of benefit rather than stuff you have to know. So essentially your small intestine looks like this, right? And really the whole purpose of this, these are called villi, is to increase the surface area available for absorption of nutrients. Actually, if you think about any process essentially in biology, surface area is king, right? So if you think about absorption of oxygen from the air, your alveoli create a very large surface area over which that can happen. Same for your intestine, same for your skin for thermoregulation, right? Large surface area allows you to do stuff. But anyway, essentially down here, these are stem cells. And these cells up here, you know, there's a, you know, gut contents. These are continuously getting worn away and dying. And so, doggo, these stem cells produce new gut cells, and these basically kind of go up in a conveyor belt so that they move up the villi. Literally, they kind of get pushed up there by the continual division of cells down here to replace the ones which are lost through, you know, ablation or overuse or whatever that are at the tip of the villi. So right down in the bottom, these are called crypts, which I think is really cool. So the bottom of the villus is called the crypt, kind of like Bram Stoker type stuff. But there's a continual procession of cells up the walls of the crypt and differentiation as they go into various types of cells, like Paneth cells or whatever, or mucosa secreting cells, or, you know, I'm not a gut expert, but those cells at the top are then replaced. So even if you're just sitting on the couch doing diddly squat, you have to be constantly absorbing nutrients from your gut, or, you know, consuming nutrients and absorbing them and then metabolizing them to generate the waste heat which maintains our body temperature. So even if you're doing nothing, literally sleeping, right, which isn't nothing, it's sleeping, you know, so it has a purpose too. But even if you're physically static, you are continuously doing stuff as an organism, right? If you're sleeping, you're probably repairing synapse connections. Maybe you're making new ones. You're replenishing the neurotransmitters, right, which you're going to need throughout the day. Your gut is continuously rejuvenating itself. Your skin is constantly growing and dividing and shedding itself, because that's another surface which undergoes a lot of wear and tear, right? And so all of those processes are controlled at the level of the DNA and above to generate the proteins required for doing that. Whether that's that's why I always tired. Yeah, that's right. It's tiring doing nothing, right? Actually, you know, it's crazy. This is kind of one of my pet favorite things to talk about. But the reason why snakes eat once a month and why we eat 3 times a day is because we are warm, right? And the reason we are warm is that all the waste heat from essentially from cellular respiration is used to maintain our body temperature. So even if you're doing nothing, you can't not eat anything, because you essentially are breaking down glucose and generating ATP, even if you don't actually need the ATP to generate the heat required to maintain your body temperature. That's also why mice live for, you know, a year, 2 years maybe, and elephants can live for 100 years. Because elephants have a far, far lower surface area to their volume, and so they lose a lot less of their body heat. That's why mice are covered in fur, and they also live in colder places, and elephants are not. They live in warmer places too. So there's not like a perfect, you know, analogy. But essentially, an elephant's metabolism goes slower because it needs to generate less heat to maintain its body temperature. whereas a mouse's metabolism goes at like rocket ship levels because it loses so much heat continuously. And also it has a different kind of reproductive strategy too. You know, for mice, it's all about like just popping them out whenever you can. And some of them get eaten and some of them don't. Whereas elephants a little bit more into the whole nurture process. So that also plays into that. But a large part of it comes down to heat loss. Anyway, so that's just a tangent, right? So we got to get back on track a little bit. But yeah, so you can see why, you know, I can get so enthused about all of this, right? Because really, whatever thing you look at, anything, even if you look at, let's say, butterfly wing spots, some butterflies have them, some don't. The ones that have them have, in some cases, a selective advantage because they're less likely to be predated, right? They gained those wing spots, they think, there's a lot of contention about this, because of a gain of a regulatory element that turned on eye pigment genes in their wings just through random mutation, probably a transposon. I usually blame transposons, right? But that difference, right, has led to an entire fork in the evolution of butterflies, for example, right? And so it doesn't matter what level you look at it, what your personal particular interest is, whether it's at like the molecular and cell biology end of things, which is kind of more my cup of tea, or if you're interested in like large scale evolutionary processes. At the end of the day, it all comes down to DNA and how functional proteins are produced from that DNA. I mean, at the end of the day, that is what underlies everything we're biological, right? And so, yeah, you can see how I get all kind of crazy about it. Anyway, so we got to get back on track, otherwise we'll never get through the material we need to get through. So a lot of this is really just stuff that we've been talking about actually for quite a while. Ooh. All right, definitely better get back on track. And so what I want to give you is some kind of general terms, right? And these are terms which we're going to be using a bunch, right? And so when we talk about regulation, we're talking about the consequences of that regulation. And so if we're talking about negative regulation, we're talking about turning down or turning off transcription. And this is really about, we're really going into transcriptional regulation right now. We'll cover the more complex aspects of the rest of it, probably more in eukaryotic control of gene expression, because there are a lot more readily available examples for that. And so in negative regulation, you have a repressor, right? So this is typically another protein which prevents the transcription of that gene. So the default state of that gene transcription is on, right? So in negative regulation, transcription is on and we're turning it off, right? So that's the outcome. The outcome is negative. That's quite often the case in prokaryotes as a general rule. Not a hard and fast one, but a good general rule. In positive regulation, the default state is off, right? And so essentially we want a positive outcome from the effects of that control. And so that means the default state of transcription is off, and we need something else to turn it on. Transcription factor, transcriptional activated proteins, whatever it might be, enhancers, right? So in that case, the outcome is positive because now we have transcription. That's actually a much more common strategy in eukaryotes. Eukaryotes, generally speaking, again, not always, but generally speaking, genes are off until they're turned on. So eukaryotes are much bigger on positive regulation than prokaryotes. And this is just a nice kind of pictorial example of that, right? So, and this is essentially, we're talking about things which are upstream of the transcription start site, which is shown by this kind of red arrow. That's what that means. So from that start of that red arrow, the base, I guess, that's going to be the start of the transcript. So in terms of control of transcription, not necessarily translation, but transcription, all of the areas we're talking about, the proteins can bind to or might not bind to or whatever, right, to control transcription, are all upstream. So in this picture, to the left, actually, is that way? I don't know. I don't know which way I'm pointing at on the camera. Right, and so that's in the promoter region, right? If you remember way back when we talked about gene structure and the like, we have our open reading frame, the bit that contains the mRNA, right? Oops, go back. That's downstream of this arrow or this start site. And everything upstream before the start of transcription is called a promoter. And again, you can have core promoters and regulatory promoters, but those are all areas of DNA which proteins recognize and bind to. Right, so in this case, we have an activator binding site. That DNA is recognized by this protein as a particular shape and conformation that recognizes a sequence in that DNA, which allows it to bind, and that helps turn on transcription. That sequence is specific to that protein. So that protein will only bind DNA. There's some other caveats too, but it will only bind DNA that has that particular sequence. So now you can start thinking, oh, well, here's how we get some of that specificity, right? You can bet your bottom dollar that there are no DNA binding sites for the whatever turns on insulin expression in genes involved in neuron-specific stuff. Right, so the presence or absence of these particular DNA sequences is one of the ways in which you can control the specificity of where or when that gene is turned on. Now we also have another couple of wrinkles. Just to add to the terminology. And so these are quite straightforward, right? This is about, something binds, turns it off, something binds, turns it on. Now we can also have an extra level of complexity, which is about whether or not that expression can be induced or repressed. And again, this is the outcome of that action. So if it's inducible, then typically that's transcription is off due to the presence of something. In this case, it's called a repressor. And so the inducer induces the repressor to leave. That then turns on gene expression. So
what would be like the main difference between the inducible and the positive if they both like turn on the transcription?
So positive regulation, you require the binding of something to turn it on. In inducible, you require the removal of something to turn it on. both of them are the same in that the default state is off, but they're off for different reasons. In positive, it's off because there's no protein present that turns it on. In inducible, it's off because of the presence of a repressor which prevents it from being expressed or transcribed. And so you have to induce that repressor to leave for transcription to occur. Good question, though. So this is essentially getting some of that complexity of what's going on, right? So it's not just about stuff binding to things to turn them on or binding to them to turn them off, but also getting rid of those things or inducing those things to leave or creating a situation in which those things bind. And so similarly with repressible transcription, this is the same in some senses as negative regulation, right? Both have a default on state, right? And so in negative regulation, you're inducing well, essentially something has to bind to turn off gene expression. And really, repressible is actually kind of the same thing in many ways, right? But now we're talking about the actual actor involved. And the extra wrinkle here is it's not just the binding of a protein. It has to be the binding of a protein and something else, right? So now we're talking about a repressor, right? Because it's repressible. But that repressor only functions in the presence of a co-repressor. This is very much the case in, oh, I've got to be careful, make sure I don't say things wrong. I'll talk about it later. Don't want to confuse matters. And so in this system, in repressible transcription, we have two things that are required for transcription to be turned off. We need our aporepressor, right? And so, where's the chat box gone? So the aporepressor, oops, only one P. So the aporepressor is something that is able to bind DNA to repress transcription, but can only do so when it's joined together with its co-repressor, right? And so the co-repressor can be many different things. Oh, did he bring you a drink?
Yes, he's sick, so he couldn't go to school today.
Oh,
He's my 10-year-old. He's my oldest.
So a co-repressor can be lots of different things, but especially in prokaryotes at least, they're often metabolites. So if you think about the purpose of producing a metabolic pathway is to generate a whole bunch of metabolite that you want, let's say. Well, once you have a lot of that, you don't necessarily need that metabolic pathway to be expressed at such high levels. And so quite often co-repressors are the metabolites that are the product of the pathway that they kind of help to replace. So it's kind of like that feedback loop, essentially. And so this is just a kind of a pictorial description of what we're talking about. So if you're inducing, you're essentially inducing a repressor to leave. That's definitely the case in the control of the lac operon, which is what we'll cover next. So the inducer is lactose, right? It's the metabolite that's going to be, this is substrate, pardon me, of the products of the lac operon, right? And so once this inducer is present, it binds to the repressor and essentially changes the shape of the repressor such that it isn't able to bind DNA anymore. And that gets it out of the way, literally, because it's often a physical block on transcription, and transcription is allowed to occur. The opposite being the case here, right? Here's that our aporepressor, you note that in the absence of the co-repressor, it doesn't bind, right? So it needs that co-repressor present to adopt the correct shape so that it recognizes the DNA and then it binds the DNA upstream of the transcription start site and blocks transcription. These are particularly common in prokaryotes, in bacteria. And so this last point here, and genes can be controlled in many different ways by many different things, right? It's not just a one gene, one protein kind of deal, right? You can get many repressors and inducers and activators of gene expression. But one of the particularly fun things, and this is especially common in prokaryotes are, well, I'd say very parsimonious, right? And parsimonious means being very careful with what you have, right? So bacteria are constantly in competition with essentially their clones and also other microorganisms, other bacteria present in their ecological niche. So they are very good at taking advantage of any new metabolite that comes along that might give them the edge, but also not expressing any genes any more than they absolutely have to. They are very careful with their money, if you want to think of it in that sense. That's actually one way in which prokaryotes are really cool to study in terms of control of gene expression. It's almost what one could say is exquisite, right? How carefully and how precisely they control their response to their environment. Okay, so a couple of key points, and then we're going to introduce the lac operon, but then we'll probably call it a day and leave it there, right? Because I know from experience that lac operon isn't too bad, but the trip operon is a real challenge for a lot of students. So I want to make sure that we go through it kind of in a measured manner. So most of the control of gene expression in prokaryotes is done through control of transcription. But don't forget, what is one of the neat things about transcription and translation in prokaryotes? This is a question in the exam.
It can be done at the same time.
That's right, they're coupled. And this is an utterly essential part of some of how they control the expression of those genes. Also, bacterial mRNA is polycistronic, right? So typically, all of the genes that are involved in a particular metabolic pathway are grouped together in one mRNA transcript. And thus the expression of those different genes is all controlled together by one single regulatory region. This is called coordinate regulation, right? So this is how bacteria control the expression of genes involved in the same process, right? When more than one gene is required. And also this whole thing together is called an operon, right? So an operon is basically a series of genes grouped together, which are going to make your polycistronic mRNA. And there's going to be a series of regulatory sequences that are used to control that production of that polycistronic mRNA. This is one really big difference between prokaryotes and eukaryotes. In eukaryotes, those genes that are involved in the same pathway could be on potentially different chromosomes entirely. So that eukaryotes use other mechanisms to coordinate the regulation of those multiple genes. And so another thing that I want to get across is like a real core principle in prokaryotes. Essentially, in prokaryotes, you have two aspects to control of gene expression. You have like this big on-off switch, you know, like in not Dracula, Frankenstein's monster, you know, that giant kind of blade switch that they use to kind of electrify the corpse and generate Frankenstein's monster, right? You have one of those. So you have one main kind of, is it on or is it off switch, right? Which controls the level of transcription potentially thousandfold, right? And also you have a, you know, some small nondescript guy in the corner, you know, wearing his like work hat or something with a dial, right? So basically fine tuning the level of transcription from that gonzo on or off. And so both the examples we're going to look at, the lac operon and the trip operon, both use that. That's often called attenuation because you're attenuating or reducing slightly. Sorry, distracted by cat. The level of transcription. Okay. So keep those in mind, right? Because we're going to come back to that for both of these examples. And it's really, really helpful in understanding like the overall big picture of what's going on. And if you think about the big picture as well, quite often it's a matter of, you know, say you're on a new substrate, something you can use for nutrition, right? Then you're going to go, whoa, gonzo, right? Like, gobble all this stuff up. Actually, it's almost like having dinner, something particularly tasty when you go to a restaurant. You get all excited, you kind of like eat like crazy. And then as you start getting full, you're like, oh, I'm just going to like slow down a bit and just enjoy this a little bit more. And then dessert, obviously, because you can't not have dessert. But then at some point, you're going to be fairly comatose and you're not going to want to eat anymore, right? So now you're kind of essentially you're attenuating your consumption of whatever delicious food it is that you're eating, right? And so that's essentially what bacteria are doing. They're getting what they need, but once they've got most of what they need, then they're going to dial back the expression of these genes because they don't need as much anymore, right? And so if you produce more than what you need in terms of protein, that has a metabolic cost, right? And if your buddies next to you are not doing that, they're going to outgrow and outcompete you, essentially. That's kind of like the ecological picture. And so to expand on this whole operon bit a little bit more, actually, we're probably not going to get to the lac operon much, but we'll see. Essentially, you have your operon, right? And so operon are all of the structural genes and the regulatory region to control the expression of that. And so you can have multiple, like trip-operon has 5, I think, from memory. There's not really any particular limit on that. And you'll have 2 things. You'll have a promoter. This is where, pardon me, RNA polymerase binds, typically. And you'll have an operator. And so the operator is between where RNA polymerase binds and the start of transcription. So now you can see if there's anything bound in the operator, RNA polymerase is not going to get through. So this is a blockage point, essentially, for the control of expression. This operator is very important. And this is all together. Now, separately, you have another region which is typically involved in the production of the regulator protein. This is not necessarily right next to this area because it doesn't have to be, right? The purpose of this region is to produce a regulator protein which binds or doesn't bind, depending on conditions, the operator of that operon. This is produced elsewhere. It's obviously on the same chromosome because it's bacteria, but it could be way off in the boonies somewhere else. And so all of these things together constitute the control of that operon. Right? You have your regulator, which produces a protein. You have your promoter and operator, which determine whether or not RNA polymerase is able to bind to and then process through and transcribe the structural genes, that polycistronic mRNA. Hello, beauty. Now, there's a lot of stuff going on here, but when you break it down, it's relatively straightforward, right? Again, these things look phenomenally complex when you look at them like wide-eyed, but when you break them down into the individual processes, it's not too bad. Don't think Tanya believes me.
So the operator is not going to let the mRNA bind, so it's going to like stop it from transcribing?
Way back a little bit. So the operator is just a piece of DNA. It's a DNA sequence. That DNA sequence can be recognized by a regulatory protein. Now, if that regulatory protein is bound to that operator, it prevents RNA polymerase from processing through the operator to start producing the polycistronic mRNA of the structural genes. So you can see the operators, basically this is where the RNA polymerase binds, right? And so it has to go through, oh, don't lick my feet. Sorry, dog's licking my toes. The RNA polymerase has to get through that operator before it can get to the start of transcription. Right? And so this region is not, this operator is not transcribed. It is purely a section of DNA which is recognized by the regulator, depending on conditions. And if that regulator is bound there, and these overlap too, right? If the regulator is bound, regulatory protein, sorry, is bound there, sometimes it prevents RNA polymerase from binding at all, right? So it doesn't allow the complex to form. Other times it physically blocks the passage of the RNA polymerase over that bit of DNA to get to the start of transcription. So if you have a regulatory protein bound here, you don't get any of this because transcription cannot occur. So it's really important to remember that all these regulatory sequences They're just bits of DNA, right? They don't do anything in and of themselves. What they are things that are recognized by other proteins, which may promote or induce, no, just promote transcription or be involved in repressing transcription.
Okay, so the operator is just like saving the spot and seeing like, hey, want to come here, like do more or not do, or
Right.
kind of like that? Okay.
Yeah. So essentially, and this is why in prokaryotes, we generally have negative regulation, right? So if nothing is present on that operator, this gene will get expressed with a few caveats, right? But generally speaking, in the absence of anything here, RNA polymerase will bind and it will transcribe that mRNA. So to turn it off, now you have to have something binding to that operator, which prevents transcription.
Okay, and that's going to be like the regulator protein.
That's going to be this thing, correct?
Okay.
Cool. I think that's enough for one day. I need some coffee. Okay, as always, and this isn't anything particularly new, I strongly suggest going through the rest of this PowerPoint before Thursday, just so you're a little bit kind of heads up on some of this stuff, right? Because these things can get confusing. All right. And I don't want you to be confused. I want you to understand what's going on because this is neat. So read through this stuff. Even if you don't fully understand it, that's fine. It will free up a little bit of space for you to process it better when we talk about it on Thursday. All right. Okay, cool beans, everybody. I'll either see you on Wednesday or I will see you on Thursday. And Tanya, did you get my response?
Yes, can we go over, because I'm pretty sure I got one correct that it's marked wrong. I'm like 99% sure.
Are you free now?
Yes.
Okay, I'm going to end this and then I'm going to send you another invite, okay?
Okay.